We are rolling. And so let's go ahead and get back to the presentation. And okay, and that's it. Back up a little bit. And yeah, so let's get to talking about sponges. The first group that we're going to talk about of the actual kingdom Animalia. So after a week, we've finally gotten onto invertebrates. They're actually now considered not to be a true phylum. They're considered to be a paraphyletic group. And if you remember, a paraphyletic group is one that contains some descendants of a common ancestor, but not all of them, in which one branch of the descendants has been arbitrarily excluded. I'll lecture about them as if they were a phylum, but there's actually some controversy as to exactly how you're supposed to classify critters at the very base of the animal tree. There's about 5,000 known species of sponge, including a few that are freshwater. I don't always have much luck in finding them when I want them, but there are some you could find in places like under rocks at one end of Lake Beaver Fork is supposed to be pretty good for freshwater sponges. So we get them in Arkansas, but the great majority of sponges are marine. And as Stephen Wright used to say, you know, imagine how deep the ocean would be if there weren't all those sponges in it. And anyway, you get them from the intertidal zone, which is the part that's exposed at high tide, all the way down to over 29,000 feet, almost as deep as you can get in the ocean. They're all benthic, which means they live on the bottom. That's a good word to know. I'm going to be using that again. When we talk about marine life, planktic or organisms, planktic or plankton, are organisms that drift and can't swim strongly enough to hold position in the water. Nekton or nectonic organisms are the ones that can swim strongly enough that they can determine where they're going. The benthos or benthic organisms are the ones that dwell on the bottom. And these, instead of being mobile benthos, are sessile, meaning they are pretty much permanently attached to a substrate. There are actually some that are capable of extremely slow crawling. One sponge in a tank was described about something like 25 years ago as being capable of crawling over the tank glass at the lightning speed of 4 millimeters every day. The sponge that did this was, of course, promptly nicknamed Speedy Gonzales. But yeah, for all practical purposes, sponges are sessile. There's at least a dozen known carnivorous sponges, and if not today, then on Monday I'll show you what those look like. But almost all sponges are filter feeders. They feed by simply pumping ridiculous amounts of water through their bodies and filtering out bacteria and tiny bits of organic matter and expelling the filtered water. They are filter feeders par excellence. What drives the pump is choanocytes, and these are these cells that look like choanoflagellates. They've got these flagella that beat and filter food particles out of the stream of water passing through the collar of microvilli. And then the choanocytes pass food particles on to other cells. There's no circulatory system. Cells just kind of pass things around. The water in a typical sponge is pumped in through very small holes called the ostia, and it's filtered inside the sponge and then passes out through one or more bigger holes called the oscula, singular osculum. So there's a couple of sponge choanocytes, and you ought to see that they're just like choanoflagellates. They've got kind of a ovalish cell body, and then that collar of microvilli. And then certainly in the top one, you can see the flagellum sticking out from inside the collar. If we zoom in on those microvilli, you can see that they're covered with a layer of sticky slime. The word for a layer of sticky slime is a glycocalyx. And you can see it actually forms these webs between the microvilli, and it improves the cell's ability to trap particles. And sponges consist of roughly 200 cell types. Sponges consist of, I think, as few as 4 and possibly as many as a dozen cell types, depending on what type of sponge you've got. But they've got much less cell differentiation than you do. The outer, quote, skin of a sponge is made up of these flattened cells that fit together edge to edge, called pinacocytes. Pinax is Greek for a tablet, a flat slab of wood or stone that you could paint or carve something on was a pinax. And so pinacocytes are the cells that look like flat slabs, flat tablets. They form the outer, quote, skin, but in most sponges, the cells don't actually fit together the way that a tissue would. You don't have the cell adhesion molecules that lock the sponges together. And so technically, this is not a tissue layer. This doesn't quite count. In fact, unfortunately, I can't show this to you, but I've actually done this before. You can take a piece of a sponge and put it through a strainer, squeezing it through a piece of linen handkerchief works pretty well. And you can separate it into this cloud of little separated cells. And if you give them the right conditions, the cells will come crawling back together and reassemble a new sponge. Sponge cells are capable of independent existence in ways that other animal cells are not. This does not work with people, for example. You can't squeeze a human through a cloth filter and have the cells come back together. I've tried. It was a mess. Choanocytes, you've seen. These are the collar cells that look like the choanoflagellates. There's also these amoeba-like cells called archaeocytes. They wander through the body. They can pick up food particles and carry them to other cells. And they're also capable of differentiating into other cell types if they're needed to. So sponges can regenerate very fast. Fragments of sponge, given a chance, can just grow into new sponges. And part of that is the archaeocytes, which are a reservoir of, well, stem cells. Like stem cells in humans, they can differentiate into adult cell types if they're needed to. Not all sponges have this, but many sponges have an internal skeleton of spicules, and a spicule is just a sharp, hard little bit of mineral. So depending on the sponge, it might be silica or it might be calcium carbonate, but many sponges have what look like little shards of silica or calcium carbonate locking together to make up a skeleton. Those are the spicules, and the cells that secrete spicules are called sclerocytes. Scleros is Greek for hard. So the cells that secrete hard things are sclerocytes. Many sponges have an extracellular matrix. Some have a skeleton made up of very big collagen fibers secreted by specialized cells called collocytes. Kollos, by the way, is Greek for glue. Collagen means stuff that generates glue, because collagen is the active ingredient in things like horse hooves and horse hides and horse bones and things like that, that back in the old days used to be boiled up together and the collagen extracted to make a type of glue. It's not used much these days, but there's still a small market for what's called hide glue. Anyway, that stuff is mostly collagen, which is why collagen is named glue stuff in Greek, and collocytes are the cells that secrete collagen, the glue stuff. Some sponges have an additional protein called spongin. The bath sponges do, and bath sponges don't have spicules. I'll talk about those in a bit. That's secreted by specialized cells called lymphocytes. Some sponges have contractile cells called myocytes. And many sponges have cells that surround the pores that can expand or contract like, I guess, little, diaphragms to open up the pores or close them, and those would be the porocytes. And then sponges can reproduce sexually, so you have egg cells and sperm cells. I'll show you pictures of these in a bit. The outer surfaces of a sponge are lined by these flat pinacocytes making up a layer called the pinacoderm. Remember, pinax is Greek for a flat tablet or a flat slab. So there's the slab skin, the pinacoderm. The inner surfaces are lined with a layer of choanocytes called the choanoderm. And neither one is technically a true tissue because they're not held together by what's called a basement membrane of collagen, and the cells are not linked by gap junctions. And again, that's what makes it possible for you to break a sponge up into individual cells and have the cells just come right back together. They're not locked together in any way that can't be fairly easily broken and fixed. There is one group of sponges that does show true tissues, and I'll talk about that, and probably have to wait till Monday. But the space in between pinacoderm and choanoderm is filled with this massive collagen called the mesohyal. Meso for middle, hyle. Originally, I think that was Greek for mud, but it came to mean just matter, stuff. So the mesohyal is the middle stuff, and other cells crawl around in the mesohyal. The amoebocytes go wandering in the mesohyal, and spicules are made inside the mesohyal. And that's basic organization. You've got a pore. Those arrows showing water flow show you water going in through the pore, and the pore is held open by a porocyte that can contract to open or close. The outer surface to the right, you have these flat pinacocytes sticking together. On the left, you have that layer of choanocytes called the choanoderm. Sorry, Fern and Bootstrap don't get along, and they're currently mixing it up just off camera. If you heard any yowling, that was it. Cats, can't live with them. Anyway, and then the mesohyal is the stuff shaded gray in the middle. You can see a sclerocyte secreting a spicule sticking out down at the bottom. There's an archaeocyte with a bunch of vacuoles, a lymphocyte secreting fibers, and an egg cell or oocyte in between. So let's look at some actual pictures. That's the outside of a sponge under scanning electron microscopy, showing the pinacoderm. The pinacocytes are kind of shaped like irregular polygons, and you can kind of see it down towards the bottom, that pinacoderm is actually made up of these little polygonal shapes, almost like a soccer ball surface. Those are the individual Panaka sites. Each one of those polygons is one cell. You can see spicules sticking out. The sponges you'd use in the bath mercifully don't have these. But many sponges have spicules that function, among other things, as an anti-predator defense. Taking a bite out of a sponge for many organisms would be like taking a bite out of a nice little Jello salad mixed with fine glass shards. You know, not the sort of thing you could actually eat much of. And then there again, you've got the choanoderm lining the interior. And the mesohyl is the stuff in between. And because I don't think I've talked about symbiosis enough this week, sponges are not only made up of cells that come from the fertilized egg of another sponge, they're packed with bacterial endosymbionts. These happen to be some cyanobacteria inside the sponge tissues. There's cases where the endosymbionts outweigh the cells that came from the sponge's egg. They outweigh the sponge's own cells. So a sponge is almost not really an animal. It's more kind of like a, collective association of cells. But, even defining what the boundaries of what is and isn't the sponge gets awfully fuzzy because you have so many symbionts in there. The reason, by the way, I probably won't get to this completely until Monday, but the reason why anybody that's not a total geek might actually care about this stuff is that sponges are one of the frontiers of drug discovery. Think about it. If you live in the ocean and your maximum top speed is 4 millimeters every day, and all you can do is just sit there, well, then predators are simply going to eat you until there's nothing left. And you've got to evolve anti-predator defenses. One of them is the spicules, which can make it very uncomfortable to eat a sponge. The other big thing that tasty organisms that can't run evolve to cope with predators is chemical defenses. Certainly plants do that. There's a huge number of drugs that we've isolated from plants, which did not evolve for our benefit. They evolved because they're toxic to would-be predators. Shoot, clover leaves. Clover cells, if they detect that they're getting eaten by a caterpillar or something like that, they actually start secreting cyanide. Did you know that? I was impressed when I found that out. And it's the same thing in the marine realm. Sponges and soft corals, which can't get away from predators, many of them use spicules, not just the sponges, but some corals as well. And a lot of them use chemical defenses. And remember what the Renaissance physician Paracelsus once said, the dose makes the poison. An awful lot of poisons are medicinal if you get the dosage right, and an awful lot of drugs will kill you if you get the dosage wrong, right? Aspirin is poisonous if you take too much of it. So a lot of these toxins, which in nature would be used to keep the sponge from getting eaten by some rando fish, have drawn interest because they might be medicinal. And there's some compounds that are being looked at pretty intensely right now as things like everything from antibiotics to anti-cancer drugs. And where I was going with all of that is that it turns out that in many cases, it's not the sponge that makes the compounds, it's the endosymbionts. So the symbionts in the sponge tissue not only favor the sponge's survival, they are potentially of great benefit to humanity at the same time. And that's why people who are not just complete hopeless geeks like me need to know some of this stuff. there is benefit in knowing it. Right. Okay. Rant over. There you've got some live sponges. On the exterior, you can't even really see them, but you can see the projections where they are. You can see these little light-colored, kind of warty-looking projections on the outside of the sponges. On the tips of those, you have pores where water comes in. They're called ostia. So the water goes in at the ostia. It's filtered inside the sponge, and how that works I'll show you in a bit. The filtered water passes out of little holes called excurrent pores into that central cavity called the spongocoel. The spongocoel is what you'd see if you look down into that hole in the tops of these sponges. And the filtered water is dumped into the spongocoel and then passes out through those openings at the top that are called oscula. So the water goes in through the sides, through the little ostia, and out through the top, through that big hole called the osculum. And the osculum opens into this cavity where the filtered water dumps into, called the spongocoel. You'll see this word again too. Coel, C-O-E-L, means an open space. So the spongocoel is just the sponge's open space. And that happens to be a look down the osculum of a barrel sponge. The barrel sponge's osculum opens into the spongocoel, and of course there's a big fish in the spongocoel. There are lots of commensal animals that live in sponges. Fish might shelter in there. There are shrimp and crabs and starfish that may not be found much of anywhere else living in that spongocoel, using it for refuge and sometimes making use of the water flow. But anyway, I picked that slide because it's a nice view of the osculum into the spongocoel. You can see ostia on the outer sides and excurrent pores on the inside of the spongocoel, where the water goes in and then where it passes out. There are, however, lots of sponges that may not have that classic anatomy. This is an encrusting sponge with lots of little ostia instead of one big one. It doesn't make a big funnel or barrel or tube shape. It makes a flat, crusty shape. And it's got lots of little oscula instead of one big one. So this is Decidia chimera, which has I don't know what it's doing with this, but it can concentrate nickel from seawater, which is interesting because nickel's a pollutant, and maybe this could lead to better ways of cleaning up polluted water sources. I'm not sure if anybody's looked into that, as to why Decidia is concentrating nickel. Weird. Right. So what's in between? The simplest way you put a sponge together is called ascon organization. And ascon is not a taxon. It's not a grouping of species. It's just the name for a plan of sponge. I've colored the choanocytes kind of yellow here, and the spongocoel I've colored blue, and the pores are also blue. So water passes from the outside through those pores into the spongocoel, and what pumps the water is the beating of the choanocyte flagella. All of that water is being pumped by hundreds of thousands of little flagella, and it's filtered and then ultimately passes out the osculum up there at the top. And that's the simplest way you can build a sponge. Basically just a bag with pores in it, and the inside of the bag is lined with the choanocytes that pump the water. The problem is that Ascon sponges can't be larger than about 1 millimeter. Hi, sweetie. Because choanocytes, with their flagella that can only reach so far, they can only pump water that's close to them. And if you had a big sponge that was an Ascon, it would develop this stagnant zone, this area that you're seeing in green in the slide on the over on the picture over on the right. There'd be this big area that didn't get pumped at all, and it would just sit there. That's not efficient. All of that water in the sponge that can't be pumped, can't be filtered for food, and might even go anoxic or something like that. So Ascon sponges are very limited in how big they can get. With larger sponges, you take the basic idea of an outer layer of pinacoderm and an inner layer of choanocytes, and you start folding the choanocyte layer so that the sponge can get more complex, while at the same time, no part of the cavities of that sponge, no part of the water system is very far away from a pumping choanocyte. So this is cycon organization. The choanocytes line the insides of chambers, and the chambers empty into the spongiceal. You can see that instead of forming a single layer on the inside of the spongiceal, the choanocytes form these clumps surrounding choanocyte chambers. And so water passes from the outside through a series of canals, the incurrent canals, to reach the choanocyte chambers. And because the sponge is built that way, it can be larger. Really big sponges are put together through what's called leucon organization, where the choanocyte chambers are kind of spherical shaped, and canals bring water in, and they also carry water out. So in a cycon sponge, the choanocyte chambers empty into the spongiceal. In a leucon sponge, the choanocyte chambers empty into more canals. So there's canals going in and canals going out. And the practical upshot of this is that these sponges can get really big. The biggest sponges that I can think of, giant Caribbean barrel sponges, are big enough that scuba divers can sit comfortably inside. And what makes it possible for them to get that large is this leucon grade of organization, or because of these branching canals, you can pump a whole lot of water and all of it gets filtered. All of it passes close to a choanocyte, so all of it gets moved and pumped, and you never get a stagnant zone. You never get water that doesn't pass close to a choanocyte and that doesn't get filtered. And that's a slice through an actual leucon grade sponge. And all of those round things that I guess the big, I don't know if you can see my arrow or not. I'm not sure if my pointer works when I'm doing this, when I'm projecting. If you can't see my arrow, it's the wide open space in close to the dead center of the picture. That's a canal, but surrounding it at about 12 o'clock and 3 o'clock and 5 and 6 and 7 and 9 and 10, those are choanocyte chambers. And up at about your 12 o'clock, you can see one of those choanocyte chambers actually has a connection to a canal. Shoot me a chat message if you can see my pointer arrow, because I'm a little handicapped because I can't just walk up to the screen and point at things with my finger. Hopefully I haven't made that impossibly unclear. And here's the upshot of all of this. Sponges are great filterers because they take advantage of a property of water. Water is close enough to being incompressible. You can't compress water into a smaller space by making it flow, at least not until you're getting close to supersonic speeds. You do have to worry about the effects of water compression if you're doing something like building the propellers for a fast attack submarine that has to be silent, because you have to worry about a property called cavitation. But for the world that living things live in, not attack submarines, but life, water is incompressible. You can't pack it down into a smaller space. And that's ultimately why if you have water flowing through a pipe and the pipe gets wider, the flow slows down. And if water flows through a pipe and the pipe gets narrower, the water speeds up. You've seen this if you've ever watered a garden, right? You have water flowing through a hose. If you want the water to move faster, which means it'll go farther, right? You stick your thumb partly in the hose stream, right? You put your thumb over the outflow of the hose. So what you've done is made that water to flow through a narrower opening, and that forces the water to go faster. You're giving that water less space to move, so it has to move faster to get out of the way, because if it didn't, the water would compress on itself, and water can't do that. By the same token, if you had a garden hose and you widened it, the water would flow more slowly through the widened part. You can actually see this in things like rivers. Wide rivers tend to flow slowly, but if you've got a wide river and then it flows through a narrow canyon or something like that, the water flow speeds up, and you might get white water and, exciting, rafting adventures and things like that. On the flip side, when you have a river flowing and the river opens up into a lake, the flow in the lake slows down. That actually causes the water flowing into the lake. When it slows down, it drops its sediment, and that means that lakes will silt up if you don't do various things to keep that from happening. Something that people who design reservoirs have to worry about. But the overall principle is that the speed of a fluid in a tube is inversely proportional to the cross-sectional area. The greater the cross-sectional area of a tube, the slower the flow. And if you have a tube that branches into several tubes, the speed of the flow is proportional to the total cross-sectional area at any given point. You can actually relate this to the human circulatory system. Blood passing through your aorta is moving at, I can't remember what the arterial velocity in the aorta is, but it's several feet per second. Not that I would ever do such a thing unless you cheat on my test, but if I were to stab you in the aorta, blood could potentially spurt out several feet with every heartbeat. Again, I would not actually do that, but the blood coming right out of your heart is moving pretty fast. That doesn't happen when you get a paper cut on your finger, right? When you get a paper cut, blood does not shoot out of your finger at several feet per second, and you don't spatter the walls. Blood oozes out very, very slowly. And the reason is that blood's moving fast through your aorta, but the capillaries collectively, even though each one is just, maybe 10 microns wide, the total cross-sectional area of all of the capillaries in your body is much, much greater than the cross-sectional area of your aorta, which is roughly the size of an egg roll. So there. So the reason why blood flows very slowly in your capillaries but very fast in your aorta is that your capillaries added up all together have far greater cross-sectional area than your aorta does. And that means blood flows through your capillaries slowly. It doesn't jet out. and there's time for that blood to do gas exchange. There's time for blood to take up carbon dioxide and release oxygen to the tissues. And sponges do the same thing. Water moving into a sponge may be moving at 7 millimeters a second. That's not hugely speedy, but it's decently fast. But as it passes through those in-current canals, and as those canals branch and fork, the cross-sectional area goes up immensely. And so the total cross-sectional area of all those choanocyte chambers might be 100,000 times the cross-sectional area of all of the canals leading in. So when water hits those choanocyte chambers, it slows down a great deal, and that gives the choanocytes time to pick food particles out. And then the water speeds up again as it passes through the excurrent canals. And when it goes out of the osculum, it's moving fairly fast, which means that it will travel some distance away from the sponge, so the sponge is not filtering the same water over and over again. So very neat adaptations right there for sponge filtering abilities, taking advantage of some very basic principles of fluid dynamics, principles that are also found in the engineering of your own circulatory system. Okay, right. Probably more than you wanted to know, but again, look for the connections. There's connections everywhere to other fields of biology. Okay, most sponges produce spicules, although not all of them do. The spicules in one major group, they're made of silica. In another one, they're silica. In a third one, they're calcium carbonate. So it depends on the type of sponge. And the spicules come in many shapes. They're often quite beautifully geometric. And the shape of the spicule can be important for identifying what type of sponge you've got. The largest class of sponges produces fibers of a very tough protein called spongin, either in addition to spicules or sometimes instead of spicules. The demosponges that don't make spicules, but that do make spongin, are the ones that are actually farmed and harvested commercially for cleaning purposes. Most of the sponges you would just buy for like your kitchen sink are actually artificial. They're made out of artificial rubber or plastic or something like that. But you can get natural sponges at places like Bed Bath & Beyond. I've also seen them sold at hobby stores, you know, like Michael's and such. Usually not so much for cleaning yourself as for painting. Some painters like to use a natural sponge to dab paint on a canvas. So that's just a mixture of sponge spicules from different species. We probably won't get to do this in lab, but you can digest these out with a little bleach. And there's a bunch of names for naming different spicule types. So the ones that are just one long rays are called monaxons, which may be monactine if they have only one point, which means they look like pins, or diactine if they're pointed on either end. You can see some tetractines, the ones that look like crosses, have 4 points. And there's various others. There's a whole bunch of jargon names for different spicule shapes. Each sponge may only have a couple of spicule shapes, but again, figuring those out might be important for identifying the sponge. These happen to be some spicules that a friend of mine collected. I had a friend of mine in grad school had a business degree and was working in San Francisco in the field of business and was making good money, and decided he wanted to go back to school for his MBA. And to get admitted, he had some BS requirements that he'd never quite finished. He had to take a science class. He took a night class in paleontology, and he fell in love. And he was my fellow grad student, and he dropped his job and quit earning all that money and became an expert in sponges and jellyfish. We actually published a couple of papers together. His girlfriend left him, but he got a much better one. And then a much better one after that, and she's now his wife, and he's now a scientist at the Smithsonian. And I mention this because he took this picture in grad school with our scanning electron microscope of a sponge specimen that he'd collected on a trip that we took. We would take trips down to Baja California, Mexico, to scuba dive. Well, he did. I couldn't scuba dive, but I'd snorkel and he'd scuba dive, and we'd collect invertebrates that our friends would use in their research projects. And so he collected this sponge, and we just took it back, and we got a nice picture of the spicules. So yeah, those were the days. I had friends that would go down and do invertebrate zoology in tropical Mexico and take drugs, and then come back to California and study the spicules. And these happen to be triaxon triactines, and they show you that the sponges have two different spicule sizes. Megascleres are big and microscleres are smaller. You can see how you have these large kind of Y-shaped spicules and then smaller ones that are also shaped like Ys. The microscleres aren't necessarily the same shape as the megascleres. It just happened that way this time. There are some sponges that have very long spicules that are like fiberglass in texture. These long spicules actually have fiber optic properties. I don't know if that's adaptive or not, but you can transmit pulses of light down them. And things like this are used to anchor the sponge in soft sediments. The spicules are secreted by specialized cells called sclerocytes. Again, sclerocyte means hard cells. Scleros means hard. And they start within a specialized vacuole of the cell. The cell will first secrete a little organic matter, and then will start laying down layer upon layer of mineral surrounding the spicule. And as the spicule gets bigger, the cell may end up dividing. You may end up with a whole bunch of cells gathering together, all of them secreting fresh mineral material on top until you get a full-size spicule. And that's the truth. That's not speculation. Right. That's a slice through a sclerocyte caught in the act. That big, very dark-colored bar across the middle of the slide is the actual spicule. And you can see right there, down below, you can see the edge of the vacuole that it's forming in. And then above, you can see the nucleus of the cell that's making the spicule, the sclerocyte. As I've mentioned, bath sponges don't make spicules, and it's a good thing because they'd shred your skin if you tried to wash with one of those. You might end up terminally exfoliated. But the ones that are harvested commercially for bath sponge purposes don't make spicules, but they do have a skeleton made of this network of tough but flexible and elastic protein fibers. So if you took a piece of bath sponge from Bed Bath & Beyond and looked at it under the microscope, that's what you'd see. That's what natural sponges look like. There are demo sponges where the spicules are embedded in these fibers, but the bath sponges don't have them at all, don't have spicules at all. And let me see what time it is. It's 11:42. Do you want to keep going? Okay, Autumn, you're saying you cannot. Oh, you're saying you can't see the pointer that I'm using. All right. Okay, you can't see the pointer. All right, I will. Um, I don't know that I've got a solution for that. I'm used to being able to, physically go up and point at things on a slide to indicate exactly what on the slide I'm talking about. I'll try to make that as clear orally as I can. Okay, I could go on, but that's kind of a lot to absorb. So, by the power vested to me by the state of Arkansas, unless anybody's got any questions I need to answer, I'll go ahead and give you a couple of minutes to type if you need to. But I'm going to go ahead and cut the recording. I'm not quite done with sponges. We'll finish up with them on Monday, and then continue with some other phyla, and we'll be on our way. Okay, once I've cut the recording, I'll take the question about, what the heck. I haven't quite figured out exactly what the exam format is going to be. I've done take-home exams before, and I have to come up with ways to kind of personalize them, because I'm sure that all of you are as honest as the day is long and would not even think of committing academic dishonesty. But I am coming up on 21 years of teaching at UCA, which means about 27 years of teaching at the college level, and I am a suspicious bastard. And you can put that on the course evaluations. Just spell bastard with 2 A's, please. I'm tired of seeing it misspelled. So if I give the kind of exams that I usually do where I can't proctor, there's always that temptation, and I would not want you to give into it. I can always try giving them through Blackboard, which I hate, Twitch, but I could do it. But not everybody necessarily has access to a lockdown browser that's supposed to have some anti-cheating measures, which means I could give you guys a take-home, which, yeah, Philip is suggesting that. But I need a little time to think about exactly how I'm going to play this. If you can hang on till Monday, I'll try to explain how the exams are going to work then. Okay, right. Oh, types and formats of questions. When I can proctor, it's a mixture of short answer and essay. When I've given take-homes, I have this nasty habit of writing these questions that actually require a little bit of research to answer, and that may not. Hello, Kat. Hi, Fern. Hi. Say hi. And that may not have one fixed answer, but might require you to, instead of spitting out an answer, come up with a hypothesis or something like that. That might require you to do some background research. Some people love questions like this, and some people can't stand them. In the real world, you always can look up all of these facts that I'm spitting out in the lectures, and it's what you do with them that ultimately makes a difference. That's ultimately where the educational rubber meets the road. So philosophically, I like asking questions like that. Sometimes it's hard to write questions like that are actually fair and also rigorous, but that's probably what I'm leaning towards. And if that's the direction I go in, then you ought to have some familiarity with the vocabulary that I'm presenting, just so you know what to stick onto Wikipedia. But it's more important to get the concepts down than to know what every last little bit of every animal is called. That's the way I'm leaning. But again, I haven't quite decided what the exam format's going to be. So I will work on it this weekend and get back to you on Monday. How's that? All right. I'm going to go ahead and cut the recording now. Say goodbye, kitty cat. Bye, kitty cat.